Fluorescent protein purification.
Today you will get to do an experiment that utilizes techniques that are usually only used in laboratories. If you successfully separate fluorescent proteins from the other molecules in the bacteria, you will be a true scientist. This first step is to find your agar plate. Once you have your plate, you can begin using your transfer pipette and add 0.5 milliliters of your TE buffer onto your plate. Pipetting small amounts is not an easy task, especially when you're using disposable pipettes. To make sure you have as close to 0.5 milliliters transferred onto your agar plate, please make sure the correct amount is in your pipette so you can empty all of it onto your plate. Using the transfer loop, very gently scrape all of the bacteria off the surface. You have to be extra careful not to poke any lines in the agar. It will be as though you are scraping whipped cream off of Jell-O. Make sure you are extremely gentle. Collect all of the bacteria using the same pipette. Collect all of the scraped off bacteria and take these bacteria and put them into the clear microcentrifuge tube. To make sure you get all of your bacteria cells off your agar plate, add another 0.5 milliliters of TE buffer to the plate and scrape the remaining cells. Remember, you can't be too careful. Add these to the same clear tube. Close the tube and gently mix the solution. Using a transfer pipette, add 1 milliliter of lysozyme solution in the tube and gently mix the solution for 7 minutes by turning it up and down. The lysozyme is used to break down the bacteria cells in this tube. On the tube, label it with your initials and class period. The tube now needs to be snap frozen, or frozen overnight. Freezing the solution makes it so the bacteria cells expand and break. Now, by simply using your body heat, defrost your labeled tube in your hand. Next, you will need to place your tube in your classroom's centrifuge. Make a counterbalance for the centrifuge tube you want to put in the centrifuge. This means that the masses, not volumes of the tubes, should be as close as possible. Unbalanced tubes may permanently damage the centrifuge. Put the tubes opposite each other in the centrifuge. If you have more than 2 tubes, only the ones opposite each other have to be equal in mass. Enter your settings, such as rotations per minute. Remove the tubes carefully after the centrifuge has completely stopped spinning. This is so that the different suspensions do not mix again. Make sure your tubes are balanced with the other tubes, and then let your tubes spin for 10 minutes. Next, we're going to grab our tube and collect the supernatant. Using a new pipette that will become your transfer pipette, collect all of the supernatant. Now add the supernatant to the solution containing the nickel beads. The nickel beads bind to the histidine tag on the fluorescent proteins. You will then rock this tube back and forth for about 3 minutes. This allows the fluorescent proteins to bind to the nickel beads. Now you will start using a new transfer pipette to transfer the entire contents of the nickel bead tube into the column. Once the flow-through is complete, cap the orange tube and put it in the waste container. Now you will place a clean microtube under the column. Use the same transfer pipette and add the elution buffer into the column. This elution buffer allows the fluorescent proteins to unbind from the nickel beads and flow through. Collect your fluorescent proteins in a clear microtube. Next is the moment we've all been waiting for. Use a UV light to observe your proteins and watch them fluoresce, fluoresce, fluoresce. Mistakes are normal in experiments, so don't get discouraged if your proteins aren't glowing like the green lantern. Tune in to Day 3 to discuss the problems you may have encountered and a full explanation on the science behind our fluorescent proteins.